All right, good afternoon, everyone. We're going to go ahead and get started. My name is Dana Oleskowitz. I am the Administrative Director for the Water Management Association, and the division, Ohio Lake Management Society, is the division that's hosting today. I'm a bit distracted because I'm still letting people into the room. And so I'm just going to go ahead and hand it off. And Brad, Carter, if you want to introduce yourselves and take it from here. We are recording this and it will be posted on our webpage here in another day. It will be available to WAMO members. Thank you. And Brad?
Yeah, thanks so much for hosting us today. I really appreciate it. My name is Brad Bartelme from a company called EnviroScience. I'm an aquatic biologist, been here for about 8 years or so now, which is kind of crazy to think about. And most of my role here, wear many hats, as do most folks, but most of my central roles are involved around water quality issues. So that's kind of the little brief background, and I'll let Carter talk about what he does.
All right. How's it going, everybody? This is actually one of my first lunch and learns here with WAMO, so this is exciting. But I'm a board member of OLMS, and I've been in that position for a number of years. I'm an aquatic biologist with Aqueduct Lake and Pond Management. I've been with Aqueduct for the past 7 years, and my role here is I basically manage larger lake systems and then also work on a number of specialty projects. Aqueduct, we're mainly based in Ohio, but we also do projects in Pennsylvania, North Carolina, and Georgia. And we pride ourselves on being a full service lake and pond management company. And just excited to give this presentation today to this group.
All right. Well, then let's begin here. So essentially today we're talking about harmful algal blooms, some trends in the field, and advances in treatment technologies. It's kind of a brief overview of what we'll be talking about today. Going to kind of nerd out for a little bit here before Carter talks and talk about some cyanobacteria, or blue-green algae is another synonymous term, and some HAB, harmful algal bloom taxa. So there's cyanotoxins, the toxins that are associated with them, and problems associated with the harmful algal blooms. Management and control methods, then Carter's going to be going through some of the treatment technologies, and he'll start off talking about a case study, one of the projects we've been focused on this past summer and last year as well. So a brief introduction to the blue-green algae here, the cyanobacteria. They're an important photosynthesizing bacteria. So they're in this bacteria group, but they're grouped with true algae. So it's kind of always interesting, explaining that to people and kind of how that works. But for the sake of this group, I'll be kind of using cyanobacteria and blue-green algae back and forth. Without them, we probably wouldn't be here. Originally, they produced a lot of our oxygen supply for our atmosphere. So that's, if you've heard of like this, the old fossils, some of the iron oxide deposits from the stromatoliths and things like that, that's all the cyanobacteria producing that oxygen at the time. So they can be used as a water quality bioindicator, similar to some other algal groups as well, but essentially, if you have certain species and they're of this variety, typically you'll see them growing in these slow, stagnant waters. It's more or less an indicator of eutrophic conditions, and most of the time it might be an indicator of like high nutrients, not necessarily terrible water quality in some instances, because cyanobacteria they're everywhere, and they're there even in good conditions as well. So, but usually you'll see them bloom into something that might produce toxins, and that's kind of how we get to where we're at for talking about some of these harmful algal blooms. So there are multiple groups, and I have we'll be focusing on the cyanobacteria, but there are other harmful algae such as the diatoms, dinoflagellates. You've probably heard of like red tide and diatoms. It's pseudonychia bloom specifically, and haptophytes. Some people call those the golden algae. So there's a lot of common names and terminology that are thrown around, but today we'll be mostly focusing on cyanobacteria specifically. So some of the common harmful algal bloom genera that will be kind of thrown out here term-wise or name-wise that may produce toxins. So they're not always producing toxins, but they have that potential to produce toxins. So some of the surface scum formers, Annie, Fanny, Mike, a lot of people like to kind of throw those names out there, but Microcystis, Anabena is actually, that's actually the benthic form, and Delicospermum is the planktic form. The Delicospermum used to be it used to be Anabena, but most of the time folks still use that old name. Cylindrospermopsis, Aphanizomenon, Planktothrix, a bunch of long, interesting, funny names. There's also some species that may form free-floating mats. So Oscillatoria, Lingbia, you'll see these growing benthically, and typically they'll get air bubbles trapped underneath and float to the surface, and you'll see these free-floating mats, and they're gross and they have a potential to produce toxins. So that's another couple other genera that you'll typically see, a couple of species. And then I'm just kind of wanted to show some photos of these. We've taken these in the lab. It just kind of gives you an idea of who the major players are in this harmful algal bloom world. A lot of the time people get to see it from the macro level perspective, on the lake or anything like that. And this is what we're seeing under the microscope. So this is Microcystis aeruginosa. There are multiple species of Microcystis, but this is one of the more common ones that we see throughout Ohio, and it's everywhere, pretty much. This is Delicospermum circinale. Before it was also called Anabena. So this is what we see also in the microscope, kind of forms these, definitely different from Microcystis, forms these filaments and has these heterocytes and aconites. I don't know if my mouse will show up here if you can see it, but some of these different accessory cells that I'll kind of go over in a little bit as well, more detail. Here we have a couple of different species of Cylindrospermopsis. So this one is a Raceborskii, but you see they have they are a filamentous similar to Delicospermum, but much thinner cells, cell width and length. And then they also have this little accessory cell up here, this apical cell, kind of like almost a little candle wick, as you might call it, something like that. And then here's another species here, just a different growth form, kind of grows in this, spiral arrangement. Aphanizomenon, it's another one of the major players here, but another filamentous, does look similar, maybe you might say, to some of the other cells, but it does form these tight, fascicles, these bands here together. They kind of group together, cluster. And what you'll see from a macro perspective, it might look like blades of grass or something like that on the surface of your pond or reservoir. So yeah, you'll see this. It's actually, you could see it from a macro perspective, but this is what it looks like under the microscope. Here we have Planktothrix. So right here is a couple different filaments, and it's kind of generic, pretty generic looking species. But it does have these, you kind of notice, the photo's a little grainy one blown up, but you can kind of see some of these like aerotopes. So they can basically hold gas in those aerotopes, these gas vacuoles, and they can float to the surface and outcompete and shade other species of algae and cyanobacteria. So all of these are the, all of those species that I kind of listed here. They're also listed right here. And here are some of the toxins that they produce. So from a human health perspective, that's of course the issue, is just trying to, mitigate these toxins. And the conundrum and what's so hard about these is that, hard about monitoring and getting rid of these is just, these species can produce different toxins. So you see here that Anabaena can produce microcystin, it can produce cylindrospermopsin and anatoxin. So really it's, you kind of, when you're monitoring for these, and specifically for the toxins, you really do want to test for multiple toxins. You want to do that full panel screening for those. And then some of the health effects are listed here as well, which, bad, not great. Don't want any of those. So kind of what I went over beforehand is that they can produce a variety of toxins, hepatotoxins, neurotoxins, dermatotoxins, and you get all sorts of symptoms after the exposure. And the problem is they can mimic other illnesses and diseases. You're seeing it more often now that blooms seem to be more prevalent. So, it's tough to discern, if you're sick, it might be tough to discern, is it cyanobacteria? Is it any other illness as well? And what makes it tough as well is that, they're really acute. If you to drink it, or if you're, dog or any human, like little kid, you're out there swimming, it's pretty huge human health risk if you were to accidentally consume some of this at a high level, as long as it's producing toxin at the time. Because there's no known antidote, no real treatment, other than just treating the symptoms that you might get from poisoning. So some of the major problems with these versus other algae is that they're tough. They have these specialized cells, which I've listed down here, and it's more terminology for you, but these heterocytes, these nitrogen fixing cells. So it's helps them as the season progresses. As nitrogen becomes limited, they can fix nitrogen, atmospheric nitrogen, with these cells. So they can persist later into the season, outcompeting other species that don't have these special accessory cells. Aconites are these little resting cells. Essentially, they can go into the sediments and then come back next year. So it's essentially a way to make sure that they stay alive into the season after season, pretty much. And then the aerotopes, which I briefly spoke about, it's basically these little gas vacuoles that allow them to float to the surface. You'll see them forming scums, and that's to shade and outcompete other species. It's just an easy way for them to photosynthesize and outcompete all the other species as well. So in terms of monitoring for these and managing these, there's kind of a multi-level approach, as I kind of say above there. There's of course the monitoring effort and collection effort, and that's what you go out and collect. And then identification and enumeration is usually the step that people tend to skip here and there when they're setting up these management plans. So really it is a good idea. You can see from that list before that you might be testing for the cyanotoxins, which is great, but if you don't know what species you might be testing for the wrong ones. So in a nutshell, it's good to have that data and track that over time, because you're having an idea of your primary production that's there, and then how many cyanobacteria you have versus relative to your green algae, just to see if it's a balanced system, or if you do have those cyanobacteria, which toxins do we need to test for? So it's good to kind of have that step in line. Then, of course, follow up with appropriate cyanotoxin analysis and implementing then some management strategies. So essentially, you need all of these pieces of the puzzle, as I kind of say. These aquatic environments, everybody kind of wants to put things into a box and have it nicely arranged, but just with the biology just doesn't do that. it's a dynamic environment, and one approach or treatment won't necessarily work. You kind of have to have a multi-level management strategy then as well, for these. And the cyanobacteria are interesting because they can be a paradox almost. You can have a certain species present at high density and no toxin, and then at a low density and high toxin, and every different arrangement that you can, every combination. I've seen it all. So there was a couple photos down here, pulled these from EPA documents, and there's no toxins at this one right here, and it looks like it is really thick bloom. You would assume that there would be toxins, but no toxins detected there. And then here it looks maybe a little bit green, but there's no real surface scums or anything like that. So you might assume, oh yeah, there's less toxin, but here it was greater than 5 micrograms per liter. So really at the root of the problem, the nutrients, of course, are the cause. So they need the nitrogen, they need the phosphorus, they need all of those micronutrients to live. So eutrophication is, of course, just how much of that is contributing to the primary production. So you're getting nutrients from point sources, where maybe it's there's specific inputs from maybe like a septic tank. That it's coming from, it's leaking there. I found that point source, if you found it. And if you don't know exactly where it's coming from, it's like a non-point source. So in the case of most reservoirs in Ohio, if you're surrounded by agriculture, those fields are going to run off, nutrients will run off into the streams flow into the reservoir, and that's where you have kind of some of those non-point sources that usually persistent throughout every season, assuming you don't manage for those or have the appropriate riparian buffers or anything in place. So overall, the problem will not go away until the root issue is addressed, which is the nutrients. So in terms of management strategies, I know what I have, how do I control it? And I'll talk a little bit about this, and then I have a couple slides, but then Carter's going to be some of the main management strategies, applied management strategies. I'll talk about a lot of those. So I tell people that knowledge is key for sure in kind of knowing what your reservoir is doing. So knowing that algae, seasonal algae cycle, seen it's pretty similar in a lot of areas. And, in winter, you might have the diatoms here. In the spring and early summer, it kind of moves to a mixed assemblage. So you can see some green, some cyanobacteria, some diatoms. And as those nutrients are used up, as those nutrients become limiting in the water column, that's when it's midsummer, warmer temperatures. Excuse me, that's when the cyanobacteria tend to start proliferating at that time. And they have those special accessory cells that they can fix atmospheric nitrogen. So that's when they're in their prime. It's essentially usually in the mid to end of summer. So in terms of managing these, managing nutrient runoff, that's the name of the game, essentially, long-term wise, for sure. So your source water protection, I just kind of have a little figure here. Riparian buffers, aquatic macrophytes, floating wetlands, sediment removal in some cases. But that's if you have these plants and you have these buffers in place, it essentially filters out the nutrients for you and puts it to biomass versus going directly into the water, running off into your lake or reservoir. So some other management strategies, controlled algicide applications in some case are good short-term management strategies. And some of these, numbers that I've put out here, if things are prohibited algicide application wise, I notice is more or less for like a lot of drinking water and things, but you'll still want to know what your cell densities are regardless, just as you're applying these algicides. So it's a good good to know, once again, to use identification and enumeration as one of those steps for managing things. And I have a few other here: aerators, diffusers, phosphorus inactivation. Carter's going to go into more of that applied knowledge here. So I'm going to pass it over to you, Carter.
All right, awesome. Well, Brad, I think that was a tremendous introduction to cyanobacteria and harmful algal blooms and the issues related to them, you know, the sources of where they're coming from, and some potential management techniques. That's kind of where I'm going to focus our efforts here. We're going to discuss this case study, but you know, there's a number of control methods for cyanobacteria. You know, basically we can lump them into two general categories where we're trying to prevent their formation through nutrient mitigation. So Brad already hit on those. You know, we can do watershed management, watershed BMPs, try to slow some of that external nutrient loading. We can also address internal nutrient loading sources, so phosphorus that's already in that lake ecosystem, either through diffused bottom aeration, which cuts off the internal loading cycle. And then we also can use, you know, some flocculants. So we have alum, aluminum sulfate. There's some technologies out there with lanthanum as well, Foslock, and then some newer technologies called Utrafix, which is also another aluminum product. So we've done a lot of work with those, but today, particularly what we're going to talk about is a case study at Roaming Shores where we're doing somewhat of a reactive management strategy. So we explored doing nutrient mitigation. It comes with extremely high price tag, you know, lake association. Basically, a lot of lake associations across the country really aren't in a situation financially where they can properly address the sources of their problems. So we're somewhat shifting towards a reactive strategy or an applied lake management strategy, where we're actually treating for these cyanobacteria blooms when they form. So in this particular case study we're going to focus on here today, it's a 450-acre recreational reservoir in Northeast Ohio in Ashtabula County. We have conducted applications of a floatable copper-based algicides called Vodagard C in the summers of 2021 and 2022. Prior to us coming on board with this project, I believe in 2018, they had a large cyanobacteria bloom that closed the lake over the 4th of July, which stemmed, you know, a lot of these, you know, seeking out additional managements for these problems. All right, next slide. So like I said, this is a applied lake management project. So we want to be out there collecting as much data as we can to help us, you know, decide what we're going to do, timing and strategy. So that's the constant battle with this type of management strategy, is going out there, collecting your data, getting it back, processing it quickly enough so you can react and implement a management technique. So in this particular scenario, EnviroScience is basically the third-party consultant out there collecting a lot of the environmental data. This is some of their work here. Basically, we have 7 water quality monitoring stations. This is from 2021, and these were shifted a little bit in 2022 based on what we were seeing. As you can see here, we have a couple lake locations that are key that you'll see further on. So we have a dam water quality sampling location, then also a mid-lake. In 2022, this was moved further down the lake to the south. So next slide. So I know we've hammered this home pretty well, but you know, nutrients are really the driving force in the blooms here at Roaming Shores as well. I don't know how many people are familiar with Liebig's law of growth, but basically the growth rate of anything is determined by the nutrient that is least abundant relative to metabolic need. So typically in the freshwater environment, that ends up being phosphorus. In some scenarios, it is nitrogen, but typically over 90% of the time, you're limiting nutrient in the freshwater environment. It's going to be total phosphorus. So when we're looking at total phosphorus readings in lake ecosystems, especially in Ohio, once we get total phosphorus readings above 50 micrograms per liter is when we're really going to start to see, these predominant blooms taking off. And then also we want to be looking at our N to P ratios. So your typical algal cell is between 5 to 7 to 1 N to P ratio of algal biomass. As Brad indicated before, a lot of these trouble algae species we're seeing are also nitrogen fixers. So they can have a competitive advantage if your N to P ratio falls beneath 64 to 1. If it's above 65 to 1 or above 100 to 1, it selects against these HABs because your green algae and other beneficial algae species actually, I wouldn't say they have an advantage, but they definitely can compete much better. So if we look at this data from 2021 on roaming shores here, we can see that basically we're right in this zone where we're just going to see annual HABs year after year unless something is done. Next slide. Okay, getting into the strategy of what we were trying to implement here. So we selected a floatable copper sulfate based algicide, Vodagard C. We had done some work with it. We've been working with the product since 2018. We've had very good results with it. We like the product because it's somewhat innovative because it allows us to apply this algicide right to the water surface. It remains there in the top 5 feet of that water column for the first 48 hours after application. So it gives us increased contact and exposure time. And we're not really treating areas of the water body that we don't have to. So it allows us to go at lower dosage rates and really get that better contact and exposure times. In this particular project, we had to do a 2-part treatment method. So basically, we were treating the northern end of the lake, waiting 2 weeks, coming back, treating the southern end of the lake when we were seeing those cell counts basically start to spike with cyanobacteria. Next slide. So we can see here, this is just kind of a quick and easy breakdown. We worked with Ohio EPA to set up a lot of these dosing rates and as well as treatment areas. We had to meet some regulations with regard to the mixing zone once the overflow goes into Rock Creek. So basically within the first 11 hours of the application, we dropped down below the mixing zone maximum for copper for Rock Creek. So we did a 500-foot setback from the dam, which is double what the manufacturer recommends for copper sensitive areas. And then, like I said before, then we cut it in half and split it up into the 2 zones 2 weeks apart. So just to be clear here, I threw this slide in here. It's kind of a simple one, but year 1, we were seeing cyanobacteria cell counts spike early June. So we did a June 15th application, then a June 28th application. And then similarly, in 2022, we thought we weren't going to see necessarily the same thing because things weren't looking that way early in the spring. But by the time it got to mid-June, we were back actually in a very similar place. And we conducted applications similarly in 2022. So here I wanted to just feature kind of the, this is truly somewhat of an innovative product here. So this is after the product is applied. What it is a copper compound that's coated with a lipid. So it allows it to remain buoyant for 48 hours after it's applied to the water column. You can see here. Okay, I had a question here. Yep, so this project in 2022 was a question about costs. So basically their annual budget for these types of applications are right around $24,000 a year. And we did explore nutrient mitigation options, and that cost was around $4 million estimated. So you can kind of see why we're doing these types of treatment strategies. But you can see here, this is the product after it's applied. We apply in windrows, basically moving down the lake. We like to apply them so the wind is basically perpendicular to the direction of the wind that day. It's somewhat important to look at that to get even distribution, and it's pretty awesome to watch. So it'll basically work its way along, and here we have kind of a video here. I know it's kind of glitchy, but when we practice this the second time through, it kind of showed a little bit better. But basically, it provides a wall or a section of control of cyanobacteria that moves throughout the lake. Yeah. So it provides tremendous control. Brad, I don't know if you want to take over here, but I think that's where we're supposed to switch, right?
Yeah. It doesn't really matter, I guess. Suppose it's kind of it's a good segue, seeing that treatment. We're looking at the chlorophyll A values here. So the red lines here are the dates of the treatments, and each one of these lines is a location, and I connected them here. Looking at it, probably should have kept the dates separate, but essentially what we're looking at is post-treatment, you see a decrease. Now here in 2022, there was a lag so with all of this field stuff, coordinating field work and everything like that, we weren't able to get out pre-application to grab a sample, or we were able to get a sample, but the chlorophyll A value just wasn't there. So you don't see it there. You kind of see that post-treatment numbers essentially though of 2021. So lower and even decreasing lower post-treatment. So the chlorophyll A values are here on the Y, and the X is the date. But what we're looking for is the threshold, or the ideal value is around 14 parts per billion chlorophyll A for inland lakes. And so we're meeting those criteria now post-application and then throughout the rest of the season. So it's been really effective treatment over time. Looking at the cell densities and cell counts, I've broken this down by just algal group versus seeing all the species. We have all the species data, but your eyes just get cross-eyed looking at it most of the time. So I broke it down by algal group, and you can see the drastic amount of blue-green algae there was in the beginning of June. So we're at that 169,463 cells per milliliter. And that just dwarfs every other algal group. So you can see post-application then dramatically decreases. And we see that that's at the dam location, mid-lake, very similar story. We do see a slight uptick of the cyanobacteria again in August, but relatively speaking, you're always going to have a mixed algal assemblage is what we're shooting for, and that's what we have here. It's maybe skewed a little bit, to cyanobacteria here at the end, or a little bit more, but generally speaking, it's a lot less than what we had in the beginning of the season. So very effective treatment. This is the dam in 2022. We did decrease, since we've, budgets and everything like that. We're always kind of trying to figure out where we can save a little cost here and there. So there is, we did counts at the beginning of the summer and then the end of the summer as well, at more locations as well. So we added one more. So In this case, this is at the dam. Once again, very similar to 2021. We're seeing a major decrease. And you're seeing a more balanced assemblage at the throughout the end of the year or the end of the season. Mid-Lake, same story. And this was so this LRR South, this is at the southernmost reach of Lake Roaming Rock, and that's where Rock Creek comes in. So that's where you have your inflow of Rock Creek. So we're seeing different algae. I mean, that's that flow from the river. You're seeing that effect. So what we're seeing here is, different algal assemblage. We're seeing some more diatoms near this river input, seeing some more green algae. And here we have much less as well, though. So in this case, you're still seeing a decrease over time, but there is much less. So definitely look at the scale here. We're still only at that 6,000 cell range, where some of the other sites are at almost 300,000. So this inflow does provide some. It was treated, and also provides some buffering capacity in terms of the flows just moving, and kind of it's less stagnant, so you don't have maybe as many number wise or cell count wise. So the phytoplankton, they're still largely dominated by the blue-green algae, and here's some of the names here. Just our Microcystis, Aphanizomenon, Warnachinia, and Aphanocapsa were the most dominant taxa among the sites. So that's what we have here. And then the cyanotoxin levels were generally really good. We did have this one value right here that did, I think it was a more or less an anomaly. I think it was based on just the sampling technique that was used, or something was wrong about this this date. But most of the time here, and this is for 2021, it was meeting recreational standards and below recreational standards. So we're looking at that 8 parts per billion for microcystin, and we're below that. And then for 2022, I was going to include some of those results, just didn't have as much time since we're still kind of putting the final report together. But still they met all of those standards this year. Didn't have to close beaches or anything like that.
Yeah, the cyanotoxin data is truly impressive. There is some other data in prior years, but 2022 was basically the first year. Most of their samples were no detect, and then also there were no hits that would require any sort of advisory level on any of the beaches or the lake or recreational use. So needless to say, the association and the residents out there were extremely happy with this past year, and provided for great recreation throughout the entire season. I guess this is my part.
Yes, yeah. Yeah, you're good.
Take right off. So some other things we're looking at. This is a newer technology, and we're really think we have a really good method of using it, and we're seeing tremendous results on these algae and things like that. But then we also want to look a little deeper, make sure we're not impacting non-target organisms or something that we're throwing out of balance that we really shouldn't be. So one of the things we're looking at is we're also doing some copper sampling in sediments. Here's the 2021 data. Basically, we're ranging between 20 and 39 milligrams per kilogram copper in sediment. Again, these are pretty low values, not really affecting any sort of inverts or anything that is living down there at these levels. And then also another important criteria is we want to make sure we're not, creating some sort of issue in case the lake association, they're actively dredging actually, but they, every lake basically needs to dredge at some point. And they want to make sure they're not creating some sort of copper issue when they need to do that type of management. So we are looking at this at multiple locations and keeping our eye on this as well. Next slide. So another thing we want to look at, so we're applying a copper algicide to, the upper waters of basically the entire lake system. We want to make sure we're not negatively impacting zooplankton species. So Brad and his crew has been collecting zooplankton samples and quantifying them and enumerating them over the past 2 years. It's following somewhat of a typical pattern where we're seeing a lot of small-bodied zooplankton, a lot of rotifers earlier in the year, and then later in the year, it kind of has transitioned to larger-bodied Daphnia and species like that. Fortunately, we're not seeing anything that would deviate from what we would normally see in a lake community that we can tell anyways, at least through the first couple years. And then we're also, we didn't put a slide in here, but we're also doing some fisheries studies out there as well to, help improve the fishery in the lake for the lake association and make sure we're not having sort of any negative impacts there as well. So
yeah, I just want to add that this is far from all of the data, that's for sure. I know we've collected a lot of parameters at many sites. So this isn't everything, that's for sure.
So yeah, I guess this is my slide again. So in conclusion, Lake Romeo Rock is not alone here, as many of us know as water resource managers. A lot of our lakes and reservoirs and water bodies are struggling with cyanobacteria issues. We just wanted to kind of highlight this case study as a potential new technology that we've seen work and seems to have a pretty good upside to it in terms of getting, season-long control of, toxic cyanobacteria blooms, getting them as they're taking off before they, have a chance to really cause issues. And moving forward, we're going to continue this project with the water quality monitoring efforts. A lot of the data you saw collected here today, we're going to continue to collect into 2023. And then, we're going to continue probably doing the algae treatments as needed. Again, the monitoring is going to be key as to, how we continue to use that. Sediment, copper sampling as well. And then long-term, we have goals of instilling some of these nutrient mitigation practices. Obviously, pursuing grant funding is going to be a big part of that. And, trying to get some of these phosphorus innovation technologies employed out there as well.
Yeah, so, just to add to that, there's a lot that really did go into this. And we're looking at it from, short-term management into long-term. A lot of that comes down to looking at the hard and fast costs of a lot of this. So certainly there's always some sort of price to it to have that water quality. And I think for a lot of folks, though, it's in a way priceless. So you can actually use your reservoir. You can actually get out there and recreate. So it's been great working with the team that we have right now, Aqueduct and folks at EnviroScience, and definitely the lake as well has been the Lake Association has been very active as well. So it's been just a good team overall. So with that, I see, I don't know if we have any questions here. If I do see, maybe bring up the chat here.
Okay, we got a lot of stuff here. Okay, the first one, does a single cyanobacteria species that produces several different cyanotoxins produce all of these cyanotoxins at the same time, or are they produced only when the cell ruptures?
So breaking that question down, they have the potential to produce multiple toxins. They do, yes, have the potential to produce multiple at the same time, but generally we'll see one will be more dominant. So the organism has to put that effort to make that toxin. And usually we'll see one dominate over another. And most of the time it might be there, but it's in a, it's in the, like a almost no detect level for some of those. And then they are held, the toxin's held internally. So when the cell ruptures, it does release that toxin. And that's why some of these algicide applications, for example, where we want to monitor, you want to know what's there, and you want to know at what level it is there. So most of the time when we're doing these applications, if it's at a high level, we don't want people on the water recreating. We want to wait till that cyanotoxin level, if it is producing a large amount of toxin at the time, you want that to photodegrade, and usually that's a couple days. So it's not really persistent, but you just want to make sure you do take the right action, right measures for when you are treating these.
So yeah, unfortunately, we haven't really run into this problem. All our treatments have been conducted when we're seeing virtually no toxins. So that's good. Next question, I think I'll take this one. Did
Sure.
I understand correctly the 14 parts per billion is the baseline value for triggering treatments? No, the 14 parts per billion is what our target is. So that's where we're going for post-treatment, ideally, with the chlorophyll A value.
Yeah, it's from a Ohio EPA inland lakes document. They sampled a whole bunch of lakes, hundreds of lakes, maybe even thousands, but and they kind of came up with that value based on the ecoregion and the size of the lake and everything like that. So that's the goal, is to meet those sort of criteria for chlorophyll A.
Okay, floating treatments. Does the residual treatment or resultant product accumulate? Does it have potential to accumulate onshore? Are there groundwater source risks for people with shallow jet wells? And if we eventually dredge, would we now have a new problem to mitigate by copper release? I could take this one as well. So the treatment, basically, as most copper-based algicides, we find that the most of the material ends up in the sediments at the bottom of the lake. Some of it does get transported downstream, but that basically accounts for all of it. As far as a groundwater risk, I don't believe that is the case. On this particular lake, there's no direct intakes from the lake itself. So that's probably not an issue. I believe they also do have municipal water in this particular lake community. And when they dredge, is copper an issue? Yes, when you dredge in Ohio and most places, you have to test for the amount of copper in your sediments before you're allowed to dispose of it. If it exceeds a threshold, then it needs to be treated as if you have a heavy metal in there, and it needs probably to be landfilled at that point. But we're nowhere near that threshold in this scenario. And next question, along the same lines as the previous question, do cyanotoxins accumulate in sediment? Brad, I'll defer to you on that one.
No, they do not. So I would say they're going to degrade. They're usually photodegrading relatively rapidly. I would say, within a week, they're gone. Within a couple days in most instances. And I wouldn't quote me, because it does depend on it depends on a lot of things. You can't really put it in a box necessarily, as I like to say, but it's you have to take multiple factors into consideration: the light level, flows, everything, in order for you to kind of test that cyanotoxin. But they would not accumulate in the sediments. Yeah. They would degrade by then.
Yeah, they can decompose. Basically, bacteria can also, other forms of bacteria can break them down too, I believe. Like if they do end up in the sediments where there's no sunlight, they'll just be decomposed at that point.
Yeah,
they're all organic compounds.
Carter, this might be
Okay,
So-
you mentioned some other nutrient mitigation strategies to compare costs. What are some of those? So basically, I quoted, a large dollar amount for a nutrient mitigation project that we're looking at. Basically, we're using a combination of alum and lanthanum to do applications on the lake to, strip the water column to a desired phosphorus level. Our target on this particular case would be like 30 micrograms per liter, and then use lanthanum to come in and bind up sediment-based phosphorus to prevent internal nutrient loading. So those are the two main technologies we're looking at with this particular project. And then, beyond that, this is a large watershed on this lake. So, any sort of watershed BMP, we're also exploring as well. We're kind of in the infancy phases of putting something together with that as well.
And that cost may seem pretty high. I mean, it is millions of dollars. That's no joke. I mean, most homeowners associations or, I mean, anybody finding funding for that's really that's an issue. But it's something, at least, this lake association, it's something you can control is your internal loading, which we're finding that there's nutrients coming from the sediments, but there's also external loading. So from the watershed, you can control one of them, and you can mitigate external loading as much as possible through other technologies as well. There's a few, I don't know if you want to mention those, Carter, but I know like Utrafix is one of the, kind of branded names, and they have different things you can inject into the water during times of highest flow events. So a lot of your nutrients are coming in, during these certain heavy rain events. That's when most of your loading is happening. So if you're treating just during that time, connect that treatment device to a level logger of the greatest inflow. In this case, it might be like Rock Creek, but other lakes, you'd have to test, know what the discharge is from those other influent streams to know where it'd be most effective. But yeah, there's a couple interesting newer technologies that folks are looking at the moment. So then we're considering as well.
All right. Well, I don't see any more questions here on the chat.
Yeah.
Crushed concrete
Yeah, thanks.
practice to reduce phosphorus loads in stream. Okay.
Yeah, thanks for sending that.
All right. Thank you.
Yep. Thanks so much, everybody.
everyone.
Appreciate it.
We'll see you at the next one.
All right. Take care.
Bye-bye. Thank you.
Yep.